Let's move on to our next speaker, Jean-Pierre Changeux from Collège de France and from the Institut Pasteur. We'll talk about genetic and epigenetic constraints on cognitive development.
Thank you very much for the invitation, because here I'm certainly the black sheep, because I'm not going to speak about language, or very little, and I am a molecular biologist in addition, which is not the tradition in this field. So I try to bridge the gap, in fact, between molecular biology and some of the data that you have already heard, and maybe we can open the discussion later on to try to see how we may bridge the levels. I wish to say that many of these ideas came back from work I did in the 70s, and it was the time of Jacques Meller, where he was believing that studying the brain was irrelevant to understand language. So we decided to do something together, and Stanislas came in the field at that time as a common bridge. So between us, yet we never published any paper together. The fact is that at that time there was some exchange between molecular biologists and linguists and so on and so forth. And I may say that unfortunately during the past decade this did not happen, at least as far as I am concerned. So I am very happy to be with you again, and to try to give you back to the old work and update it with recent developments in the field. And I wish to say that we have always been concerned by building up models of the brain as some kind of minimal representation, trying to relate specifically neural architectures, activity patterns, if possible in mathematical terms. So in all my scientific career, this has really been the motivation to try to establish theoretical models of what we were thinking about and try to test them experimentally, of course. And in particular to try to build what we called with Philippe Courrèges many years ago a formal organism, or at least part of a formal organism, which is able to, for instance, pass a given task and try to establish a causal relationship between, may say, a specific behavior or even a mental subjective process and an objective neural measurement. So this is our constant preoccupation, and I may say we are still struggling for that, even though we have difficulties, as you may imagine. Nevertheless, I think in the field of neuroscience in particular, there are so many data and experiments done without any kind of preconceived models that things are, to my opinion, not progressing very well because of that. So this is, I may say, some kind of attitude that we have during the past decades. Now, the main idea, which perhaps you may feel philosophical, which is not new certainly, is that I have been much concerned by the issue of variance and invariance. And if you may translate it in some of the language of behavioral science, between innate or genetic and I may use the word learned, acquired, or I will introduce the word epigenetic, which is the word I have used myself. And very interestingly, when you look back to Darwin, you find indeed the issue of variation, which I think it's occurring at many levels of organization. I will mention that in a minute. And the issue of selection. And of course, for us, it's a constant issue how selection does take place, in particular within the brain. So when one is, in fact, looking at language learning, I may say that unfortunately very little reference is made to the issue of the mechanism of selection which are taking place, and my opinion is that there are, of course, reward processes which are taking place at many of these levels. Now, the interesting statement of Darwin is that the difference in mind between man and the higher animals, great as it is, certainly is one of degree and not of kind, which I like very much, even though, of course, the monkeys don't speak. But maybe it's nevertheless the issue of degree, not of kind. We have to discuss that. Now, the main issue of Darwinian kind of thinking is that there is, of course, a generator of diversity, or several generators of diversity, mechanism of selection, and also, of course, of stabilization and amplification. Now, as I said, our conviction is that there are multiple levels of variability, and all of them are nested within the human brains. I emphasize the issue of nesting and also the issue of different levels. Because when one looks at given behavioral task, even the linguistics act, I don't think anybody is asking the question, where does it take place? So most of the time it's said it is the cerebral cortex. Why? And in the cerebral cortex is not a uniform organization, far from that. So this is not question. The question is not asked. And I may say that, of course, the human brain is the product of an evolution of species. There is a variability of the genome, taking millions of years, of course, but I will come back on this issue because I think it's a very important question if one wants to assess the genetic determinants of human brain organization. Then there is, of course, another kind of evolution, which I qualified epigenetic, which is taking place during ontogenetic development, not only at the level of the development of the cellular organization with, of course, cell losses, but, of course, during synaptic development. And I will come back on this issue of epigenetic variability of connections, which may take hours, sometimes years, and which is taking place abundantly in humans. I may say already at this stage that building up a human brain takes 15 years, and since Homo sapiens appeared 200 or 100,000 years ago, most likely the lifespan of the early Homo sapiens was maybe 20 to 30 years. It means that the human species has been selected by evolution to build up its brain for at least or half of its life. It's not used more. It was not used more than maybe 10, 15 years, with a few exceptions, of course. Now there is, of course, another kind of evolution, which is the dynamics of thought, where the variability is associated mostly with the spontaneous activity of the brain and change in synaptic efficacies. So one should be very clear in trying to distinction between the connectivity and the efficacies, which is not always possible to do with brain imaging, for instance, and other techniques which are done at a more global scale. So this is taking place in the range of 100 milliseconds, which are the so-called psychological scales, and time scale. And then there is, of course, a similar change in synaptic efficacies, which are taking place during cultural evolution and social one, together with the other ones I just mentioned. So there is clearly a nesting here of evolutions that we have to clarify, with the notion also that we have several levels of organization, as I just mentioned, which are itself corresponding to a level of structure, and that one should never forget that all of our brain activities are rooted in physicochemical molecular processes here, and that in between some level where access to consciousness, for instance, is taking place, we have several of organizations that we have to take into consideration. And I may say that many of the studies which are done just don't take care of this. And if you look at the relationship between gene and language, it goes from the DNA up to, I would say, access to consciousness without any kind of hesitation. So I just want to mention these few aspects as molecular biologists, if I may say, and all the implications that may have for the understanding of all the studies being done in higher brain functions. So I will say a few words about genetics and then shift to epigenesis. And the main paradox that one is facing when we try to relate the organization of the genome with the complexity of the brain is that when you look at the number of genes, or even very closely to the gene sequence from the, I would say, from the mouse to humans, and clearly the mouse don't speak, even though they shout sometimes, or cry when they suffer, and so on. There are not only the similar number of genes, but also very small differences in sequence. And all this has been recently confirmed by the studies on human genomes and also on the monkey genome and the chimp's genome, and also in the Neanderthal genome, which has been, as you know, been recently done. So there is an ambiguously for all of us a dramatic paradox here, which is that the complexity of the brain here, I use neural number as indicator, there is a dramatic increase of complexity in the course of evolution with very little change of the genome organization. So this is, of course, creating immediately an issue in all the attempts which have been done by many groups to try to relate single gene events with brain disease. And this is one example, the case of microcephaly, which is in a beautiful reductionist kind of approach, being assigned to the mutation of gene which affect microtubule-associated protein in centrosome and affect the cell division of the neuroepithelium and therefore the number of neurons in the brain. And as you can see here, give a disease which in fact give us back to the Australopithecus. Now, there are also a lot of studies which have been done on single gene issues, like the PAX gene, the FOXP2 gene. There are plenty of studies being done on that, and many other ones. And my view is that all these studies are illusions, because, of course, if there was a one-to-one relationship between gene and disease, or between gene and function, following the old principle, one gene, one enzyme, one gene, then, of course, we would not explain the difference between mouse and humans. So the ideas we have now is that, in fact, we have to think not on single genes, but on networks of genes. And they have plenty of studies now which have been developed, in particular in the case of autism, by several groups. And we have ourselves been concerned by that, that in fact the issue of establishing a relationship between genes and networks of neurons has to be thought in terms of coherent gene groups, in addition, organized in a nested hierarchical manner. I am not going to talk about this. This would be a whole seminar, but just to mention it, that it fits with some of the recent data which have been published by those who are concerned by the relationship between human genome and the organization of the brain. And of course, one still see in a very distinguished colleague books on language and genes for language, for grammar, music, math, and so on and so forth, which of course, is completely meaningless to my opinion. I am sorry to be a bit provocative, but the interesting result by Eichler that I mentioned concerned the, as you may know, the human copy number variation, or the multi-copy genes. When I said globally, of course, the number of genes, which is very small, between 20 and 30,000, doesn't change. Of course, there is some slight variability. And this variability mostly is on the number of copies. And here you have a beautiful set of results from Eichler group. And you can see here that you may have up to 6 change in copy numbers, which have been identified in human population. So this is, again, 1,000 genome studies done recently on this issue, and you don't find them in the chimps. And you can also see that, very interestingly, if you look in red to the Asian population, in green in the European, and in blue in the Yoruba, you find exactly, or nearly exactly the same. So it means that there are really universal aspects in the case of the relationship between gene and brain organization, because most of these stratified CNVs invariants may use among human population, which are not found in apes, are associated with intellectual disability, autism, schizophrenia, epilepsy. So there is here the notion that, in fact, we may have to think about human evolution, at least from the origin, that quantitative change in the gene population coding for the organism of the brain, together, of course, with a few reorganization and perhaps some point mutation. But you may really imagine that you have a wide spectrum of mutation, de novo selected each individually in human population, may not take place in such a short time, which has been devoted to human evolution. So as a consequence of these early developments, of course, one reach the idea that there are at least some innate universal disposition, which I am going to specify, but which are very global for language. And here I just illustrate this concept of language by the work of Madame de Han Lamberts, and where you can see that in the case of forward speech compared to backward speech, you find already some dispositions which are those of the adult brain, and same thing for the connectivity. So there are many other features which are present in the newborn, which I just list here: breast sucking, recognition of human face, and so on and so forth. And this means that we have really to understand at this level, still, to my opinion, the relationship between the evolution of the human genome and this predisposition, which I may say are not well understood, even from the molecular biologist's point of view. I may say the molecular biology of these early developments has to be done in the future, and very few programs, if I see NIH or whatever, are being devoted to this issue, which, to my opinion, is absolutely crucial. Now, the main issue that I want to talk about is, of course, that there is an additional level of organization which is unique to the brain. And, of course, when one goes to from the human genome to the body phenotype, one thinks, of course, the easy relationship, which is already not well understood, between the genome and the stomach, the liver, the lung. But when it goes to the brain, there is, of course, an additional level of organization. And that's the reason why I emphasize very early on, together with with Philippe Courrèges and Antoine Danchin that all the phenomena which are taking place after birth, in particular during the growth of the brain, the postnatal growth, are absolutely crucial for the understanding of higher brain functions. First of all, you have to realize that, as you know, that the brain weight is increasing 5 times from newborn to adult, that there is an immense number of connections which are being established after birth, and there are, when you look at the developing brain for 1 second, you may find 10 million synapses being formed in the child brain. So it means that an enormous number of functions which are being established after birth are under the constraint of these epigenetic phenomena. And that's the reason why very early on with Philippe Courrèges and Antoine Danchin, we introduced this selective stabilization hypothesis, where we propose that, in fact, there is no fixed genetic determinism of the adult brain organization, but only headlines, which I illustrated before, where, in fact, one define the afferent and the target axon, for instance, and the target, which is, of course, a limited target, and that they are progressively, you may extend the number of cells in these targets and have them, in addition, nested in a repetitive manner successively. And the idea being that you have, in fact, processes of selections which are taking place where the state of activity of the target is crucial. And this is, of course, accompanied by an elimination of synapses. This was proposed and modelized as a mathematical model in 1973. And that there is, of course, basic, this is, to my opinion, a key principle, not the only one, of course, a key principle in the evolution, postnatal evolution of the brain, and that, in fact, we should specify each time it's taking place when there is the so-called learning phase which is taking place in the development of the newborn brain. Now, of course, these phenomena can persist in the adult as some kind of what we call synaptic turnover, which is also very much studied by neuroscientists. Now, when I say that there was a synaptic elimination which is taking place here, I wish to emphasize that this synaptic elimination is so important in the case of the human brain that there is, this is from the data of Utenlöscher, a decline, a very significant decline of the total number of synapses here in the cerebral cortex. And this is, of course, of great importance. It's just the envelope. So it means that throughout all these periods, starting at the time of birth up to puberty, there is this phenomenon of synaptic outgrowth and elimination, and that after, at the end, there is, in fact, more elimination than production of synapses. So this is, to my opinion, a very crucial kind of phenomenon to understand, to progress in our knowledge of, if I may say, language learning and specifying how it takes place. So this is, of course, crucial for the genesis and internalization of culture. And I will say a few words about that. Now, here, a very important issue that we mathematically proposed already quite a few years ago, if I may say, I'm sorry for that, that different learning inputs may produce different connective organization and neuronal functioning abilities, but the same behavioral abilities. It means that as a consequence of I would say, some kind of Darwinian process, we anticipate that the selection is bearing on the function, is not bearing on the precise connectivity. So we anticipate variability, and sometimes very strong, as a consequence of this selective stabilization process. And here you have the beautiful example which was produced by Leiden many years ago, but with recognizable neurons. But this is also something that have to be taken into consideration for all brain imaging studies, to my opinion. So we may discuss that with Stanislas later on. So this is, of course, an issue which has to be taken into consideration. Here it was, of course, between identical twins, and there is indeed a variability of the phenotype in identical twins. These are just experiments, and there are now at least 10 or 20 papers on this issue published recently, that this elimination of synapses during active processing of information in the brain is indeed verified in nearly all the experiments where it has been done. So this is just to, of course, very briefly, and you know all about that, the early finding of Desjardins on this issue for written language. But what if I selected this slide, is because you can see already that the importance of very long projections, which are taking place between the prefrontal and the parieto-temporal occipital cortex, which are, of course, important, as Stanislas mentioned, for highly integrative functions in the brain. Now, this is just, and I refer to the work of Stanislas, of course, for this kind of postnatal selection of cultural circuits, which you all know, and is some kind of introduction to all the work which can be done on language learning. And here, the difference between illiterate and illiterate subjects, which has been very beautifully further documented and analyzed by Stanislas' group. Now, here, how to relate all this with genes? Well, the interesting theory that Carl Scott and collaborators suggested is that, in fact, in the pathology of schizophrenia and autism, there is indeed some alteration. I don't want to say that this is the only kind of phenomenon which is taking place. There is a disrupted epigenesis in the pathology of this disease, and in particular, an alteration of synaptic elimination. The American use the word pruning. I don't like it. I am not a prune, and you are not either. So this is some kind of phenomenon which is indeed taking place. And the interesting thing is that very recent data have given credit to this, since there is, for at least some of the genes from the mTOR pathway, which is an autism-associated mutation, there is a TOR pathway, whatever it is, which is involved, which involve UBIQUITIN-PROTEASOME degradation. And very interestingly, this is for us a very important aspect of this kind of molecular studies, is that there has been some proposal for drugs acting on these pathways, which affect synapse elimination and which prevent the development of abnormal patterns of synapses in the case of autistic syndromes. Now, there are also many other models at the level of the postsynaptic membrane. Now, just a few words, no more than 5 minutes, on the access to consciousness and higher brain function. I think Stanislas already alluded to that, and this is a work that we did together, starting from 1998 after a long period of collaboration. And you may find these ideas summarized in a recent review published in Neuron. So the main ideas we have is anatomical. And the issue is to try to relate the access to consciousness, or at least what is maybe called a conscious workspace, following bars and others, to a particular connectivity of the cerebral cortex. It does not explain everything. I just want to say that, because many of the states of consciousness are under the control of the ascending brainstem pathways, of course. But this is more for access to consciousness. And if you look at Ramon y Cajal, Desjardins, you already found a considerable emphasis on this long-range connectivity, which is in fact completely forgotten by all those who are doing some analysis, network analysis using graph theory and so on. So the interesting thing is that these neurons with long-range connectivity are located in layer 2 and 3, and that these areas where the layer 2 and 3 neurons are most abundant are shown here in the prefrontal parietotemporal cingulate cortex. And this was also found by Patricia Goldman-Rakish here in the monkey, as you see. So there is a reality there. The issue, the question is, can we relate conscious access to these kind of workspace, what I would call circuits? Now, this is just to illustrate the idea that this contingent of long-range connectivity is associated with a particular complexity of these layer 2 and 3 neurons, which increase with the expansion of the prefrontal cortex. Now, just a few words about the function. This is a published work by Stanislas' team, which is to try to compare seen or not seen, or conscious or non-conscious, if you wish, perception of words using this masking task. And the interesting thing is that here you find that these prefrontal cortical areas are being excited, and if you follow by ERP, but you can do that by MEG as well, you find that there is indeed a late activation which is taking place, some kind of all-or-none ignition, which is occurring around 200 to 400 milliseconds after the start of the experiment. So this is an important criterion, one among others, for I would say access to consciousness. And you can see it's not taking place in the middle temporal cortex, and has something to do with something which is present in this global neuronal workspace circuit. Now, 2 or 3 things, and I stop. The kind of processing can be followed by many other criteria. I think you can find them in this review, and in particular the phase synchronization and large-scale integration, which can be followed by EEG. And here I take the work from initially Varela and followed up by Rodriguez and Singer to show that there is a very interesting difference between the result of these experiments between normal and schizophrenics as far as the recognition of these Mooney. Has nothing to do with the moon, but with Mr. Mooney. Mooney pictures. So there is clearly here some alteration which is taking place in the schizophrenics and concern access to consciousness. Now, if you look at autism, you find also using tractography that here there is not only some alteration, but it can be further specified in the sense that in these children there are more connections than in the normal. And that, again, this is consistent with what I said, there is an alteration of the elimination of synapses, which is taking place here at the level of long-range connectivity. So this is another strong argument about the contribution of this epigenesis of long-range connectivity in the development of cognitive function. Now, just a model to show that one may also think about representation in the brain which are not affecting the state of connection by their efficacy. And as I said before, the reward system have to be implied, to my opinion. And this is an old model that we did together with Stanislas. And just to show the variability, you can see here very interesting imaging data which show clearly some difference in the representation of house, faces, and chairs, but also, and that's extremely interesting, very significant variability in the brain imaging of different subjects. And the interesting aspect also is that it has been shown for some of them that there is a dramatic contribution of the prefrontal cortex. You saw this contribution in the Stanislas data before. And in particular for learning numbers, where the prefrontal contribution is extremely strong, even stronger than in adult learning. So this means that there might be some, it's finished, some issues for all of us as neuroscientists to go from the gene up to the cognitive functions and try to not only identify the patterns of genes, I don't say single genes, which are concerned by these different phenomena, by their epigenesis, and by the contribution of activity. This is the last slide. Just to show you the amazing finding from an unpublished work that we have submitted now, which is if you look at here at day 1 and 4, you have a fantastic activation of genes in the brain as a consequence of birth. So what's happening during birth? Simply that the fetus is no longer a fetus, it's becoming a child, but there is no change in the morphology, no, very little as a consequence. And nevertheless, it means that the state of activity is controlling the genes. And therefore that there is a dramatic epigenetic phenomenon which is taking place at this stage. So I think that we have to really think about cognitive games in brain development, including, of course, language games, and that we may view the human person as a net synthesis of invariance and variance at multiple levels of organization. Thank you very much.